Okay, before we get rolling with talking about membrane transport, there's one thing I want to revisit from last time, and it has to do with the topology of membrane vesicles when they fuse with another membrane. This just is something that screws people up royally. It's like, you tell me that something that's inside the vesicle ends up outside the cell. How the hell does that happen? I'm twisting it around in my mind. I can't see it. So there's this little video I found on YouTube that we're going to go ahead and show. It's only 13 seconds long, so it won't take a lot of the time. And I'll actually put a link to this up because I think it illustrates it pretty well. So just to set the stage here, here's a vesicle, here's the cytosol, here's the plasma membrane. So watch what happens. We got the inside stuff here, and we got the outside world there. Now the membranes are going to fuse, and what was inside went outside. Let's watch it again. So does that help a little bit in terms of visualizing it? And again, I would encourage you to go back and look at this yourself. But sometimes just seeing it in kind of a quasi 3D animation like this is a lot better than trying to turn it around in your head. The other thing to keep in mind is we were looking at what happens to something soluble that's in the lumen of a vesicle there, right? But also imagine when you've got proteins stuck in the membrane of that vesicle, which part of the protein is going to end up facing the outside of the cell and which part's going to face the inside of the cell. All right. So we want to start the discussion of transport by talking about the phenomenon of diffusion. And diffusion is something we see all the time. We experience it. When we walk into a restaurant and we smell something really good, that's diffusion of odorant molecules from the kitchen out into the dining area where they're picked up by your olfactory senses. But how does diffusion work? Well, it's all about random motion of molecules in a solution. The solution can be in a gas, in air, or it can be in a liquid, like in water. And molecules are moving randomly, and how fast they're moving is a function of what the temperature is. That is, how much kinetic energy is available to impart to these different molecules. If the distribution of the molecules is not uniform to begin with, if they're not homogeneously distributed in the volume in which they exist, then over time they're going to tend to move from where they're more concentrated to where they're less concentrated. And that's not because there's any force that's causing that to happen. There's nothing pushing or pulling them. It's simply random probability. So let's look at a little animation here. So what we've got here is some water, and the blue spheres here represent water molecules. And what I'm going to do is I'm going to add some dye to this solution by just clicking here. So I put some dye down in the corner. Now see what happens to the red dye molecules over time. We can slow this down a little bit, and we can put some more dye in here. Okay, so it starts all concentrated in one corner, but watch what happens to it over time. Notice that you don't see it tending to go back in aggregate and cram itself in that corner there. And once again, it's not because there's some force pulling it outward here, nor is it being pushed. It's just that if you're a molecule and you got all this volume to move around in, What's the probability that you'll end up going from here to here as opposed to coming all back together again? And if you want to say all you dye molecules, you get back there in that corner where you started. You concentrate yourselves. Then you have to figure out what's the probability for any one dye molecule to end up there and multiply that by all of the probabilities for each of the other individual dye molecules. And you can figure out that the chance that that's happening is extremely remote. And so these things are simply diffusing. They're moving in a random fashion, but if you say, we're going to start with you all concentrated here, then there's a much higher probability you're all going to end up somewhere out in the rest of that volume than it is you're all going to end up in the same place. And you can, in this little tool, trace what an individual molecule is doing. Just follow the bouncing ball here in this little line. And you can do this several times, and you'll see that, now granted, this is a computer simulation, but the molecules aren't following any particular path that's dictated by anything else. They're just moving around, and depending on where they happen to go at a moment, they might collide with a water molecule or another dye molecule, or they might not. So this is just an illustration of how diffusion works, and you're going to hear over and over and over in this discussion that things move from areas where the concentration is high to where they're low. And keep in mind that this is simply about diffusion. It is a random probability. It doesn't mean that things can only move in one direction. Not true at all. They can move in both directions. But if you start with a whole bunch here and very little there, and then you look at what's happening over time, things are going to tend to go from where the concentration is higher to where it's lower. And that will keep happening until you reach what? Equilibrium. And at equilibrium, everything is equal. That is, you've got an equivalent concentration in two different places. And things are still moving. They're still moving this way and they're still moving that way. But now, the probability states that the concentrations are going to stay the same. That is, they aren't going to spontaneously all go back to one place. And that's what transport is really all about in a sense. Are you looking at diffusion, or is somebody putting some energy in to do some work? All right, so that's what happened in a solution where there were no barriers. That's not the way things work in a cell. In a cell, there's barriers all over the place in the form of membranes. And the membranes, you remember, were composed of lipid bilayers that were associated with proteins. And lipid bilayers are actually very effective barriers to molecules. And if you look at different categories of molecules, small nonpolar ones, small uncharged ones, larger uncharged polar molecules, and then worst of all, the ions, the ones with charges, and how they are able to get across a barrier, you find that there's a lot of differences. Take the obvious ones here. Ions and the big polar molecules. They run into a lipid bilayer, and we're not going to see the proteins for a minute. We're going to talk about what the proteins do, but for the moment, we're just thinking about that phospholipid bilayer. If you're a charged ion and you come to a lipid bilayer, are you going to just be able to diffuse right through that sucker? No. Okay, if you're a negative ion, if you're an anion, why can't you diffuse across that thing? Think about what the membrane is made of. Phospholipids, okay. And phospholipids were amphipathic molecules, right? And what kind of charge do you find on the head groups of lots of phospholipids? You can find a positive charge there, but all of them, regardless of what their head group is, have phosphate in them. And what kind of a charge does phosphate have? Negative. So they've all got a negative charge if they're a phospholipid. And while in some cases that can be partially or even totally canceled out by a positive charge of a group like a choline, which is what you were thinking of, overall, there's a negative charge on that. And so if you're an anion and you come trucking along and you diffuse yourself right up to a membrane, what's going to happen to you? You're going to bounce back. You're going to be repelled. Negative repels negative. Now, if you're a cation, oh, you could be attracted to that. So let's talk about the cation. It's attracted charge-wise to that membrane. But what about getting in between the phospholipid head groups? What about getting across that membrane interior? Is that going to tend to happen? And why not? Okay. Okay, so why, help me understand, why doesn't that charged ion want to go live amongst the fatty acid tails in the middle of the membrane? Is it repelled by them? No, you're right. So what's keeping it on the outside of the membrane if its charge is attracted to some of the charges on the membrane and it's not being repelled by the fatty acid tails? What's keeping it outside?
Well, fatty acid doesn't have an outside charge, so it's not attracted to it.
It's not attracted to it. And what is it attracted to? You're absolutely right. What is it attracted to? Well, if it's a cation, it certainly is. And where might you find some negative charge in the environment where that ion is living? You guys are missing the obvious one. What were the little blue spheres bouncing around? Water. Water's a dipole. Every water molecule has got two partial positive charges and a partial negative charge. So water loves ions, and ions love water. And so it's all about the hydrophobic effect. It isn't so much that there's something pushing that ion away from that membrane interior. It's that it's got all kinds of wonderful electrostatic interactions going on with the water molecules. And if something, some thermal energy, whatever, started to push that ion into the interior of the membrane, the water molecules are going to say, come back, we love you. And so it's never going to go there, never going to go there. Not to mention the fact that a lot of ions have a pretty big so-called hydrodynamic radius. If you take the van der Waals radius of the ion and add to it all the water molecules that it's hanging out with, never going to fit in between those polar head groups of the phospholipids. So ion isn't going to go there. Now, think about a large uncharged polar molecule, like a sugar or a nucleotide, or nucleoside it would have to be because it's uncharged. What about that guy? It's the same story, really. It's the same story. It's polar. It wants to live with water. It doesn't want to go into that membrane interior simply because there's nothing there for it. It's like, you know, and I don't mean to insult anybody here, but why would I want to go to New Jersey, you know? Actually, there's lots of good reasons to go to New Jersey. There's some cool places there. I grew up not too far from there. But the bottom line is, it's not that there's something pushing me away, but there's just nothing attracting me there. Whereas there's other places that keep me there. And that's true for your big polar molecules. Now, when we get into the small range here, polar molecules without charges, we can talk about water. We can talk about small alcohols like ethanol and glycerol. If you do careful measurements, you'll find that they can diffuse across a lipid bilayer a little bit. Not a lot, but a little bit. They don't tend to go a lot because they got all those favorable interactions with water. They're polar, keeping them in the water phase. But if a little thermal energy happens to get them into that membrane interior, they're like, all right, you know. We're small. We can fit in between these fatty acid tails, you know. A little bit of us diffuse around here, and then maybe we can get out the other side and interact with some water some more, and that'll be wonderful. So small polar molecules like water, like small alcohols, can get through a membrane a little bit. And that actually is significant. For instance, used in drug delivery, you can create a solvent for the substance that you want to get into the bloodstream, and you put it into your formulation for your patch or whatever that you're sticking on here. And this will actually help the dissolved material to be absorbed through the skin, directly across cell membranes and get in. And that's taking advantage of the fact that some of these small uncharged polar molecules can actually get through a membrane okay. Who gets through real easily? These guys, small nonpolar molecules. Things like oxygen gas, carbon dioxide. And incidentally, we asked this once before, and somebody who is an organic chemistry genius remembered the reason that we count carbon dioxide as nonpolar. Remember, it's got two oxygens bound to a carbon, right? And oxygen is more electronegative than carbon is, so why isn't that a polar molecule? And somebody remembered last time. They cancel each other, and why do they cancel each other out? You're absolutely right. 180 degrees, it's a linear molecule. It's a linear molecule. And so carbon dioxide is still a nonpolar molecule. And it's small. Other thing that's really good getting across membranes by diffusion, we talked about steroids last time, remember? Cholesterol, ergosterol, phytosterols. Those things naturally slip in between phospholipids. They're actually part of the membrane. And there's a whole group of hormones, the steroid hormones, that are based on the same structure. They're actually derived from cholesterol and from the intermediates used to build cholesterol. Those things diffuse across lipid bilayers just fine. And in fact, they don't even need any help from proteins, unlike other types of hormones, to get across a cell membrane. They'll just diffuse right through, which makes them kind of unique, because when you get to talking about signaling later with Dr. Saxena, you'll learn all about the cell membrane surface receptors that bind to hormones, that bind to signaling molecules, and transduce the information across the membrane. In the case of steroid hormones, they don't need somebody outside the cell to grab onto. They'll diffuse right through the membrane. And so the receptors for steroid hormones are inside the cell. They're in the cytosol. They're actually soluble protein molecules. So those are about the only guys that are going to diffuse across a membrane all by themselves. Everybody else is going to need a helper. And what's the helper? It's going to be a protein, specifically some kind of integral membrane protein that is associated with the membrane. And we can be talking about the plasma membrane at the surface of the cell, or we can be talking about proteins in organelles in the interior of the cell. So there are going to be transporters both at the surface and in the interior. And most movement of solutes across membranes is going to be facilitated by some kind of a protein molecule. And there are different classes of transporters for different types of solutes. There are transporters for ions. There are transporters for sugars. There are transporters for nucleotides. And there are literally thousands of these things in the cell. And each one has its own characteristic specificity, its own characteristic mechanism for helping things to get across a lipid bilayer membrane. Water even has its own transporter. I said that water is able to diffuse across a lipid bilayer all by itself to a measurable extent. And that's been demonstrated repeatedly in laboratory experiments where you create artificial phospholipid bilayers that don't have any protein associated with them. Some water can still get from one side to another. You can tell by tagging the water radioactively and then just simply saying, do we get any radioactive water molecules over here on the far side? Yes, you do. So there's no question that water can diffuse across a lipid bilayer to some extent, but most water gets into cells through a type of protein channel called an aquaporin. And aquaporins are very widely distributed in nature. And they're represented here as being kind of a spool or a cylindrical type of protein. Now, what they really are is a structure composed of multiple subunits that forms a quaternary structure that is embedded in the membrane here. And this is actually a surface model of an aquaporin, and it shows water molecules here in red and white passing through the channel in an aquaporin to reach the other side of the membrane. And the aquaporins have a sort of a filter system built into them. Part of the quaternary and tertiary structure in here forms a narrow channel, and it's a narrow channel that will just let a water molecule go through and pretty much won't let anything else fit through there. Moreover, there are distributions of amino acid R groups that allow favorable water interactions to occur while keeping out larger, more charged species. And so the aquaporin is a type of transport protein that is specific for water. It is a passive transporter, and we'll talk about active and passive transport in a little bit. But it is simply a channel by which water molecules can diffuse spontaneously. And that means that water can come in, water can go out. Which way is it going to tend to go overall? It depends on how much stuff there is for it to interact with on either side of the membrane. If there's more dissolved stuff on one side than there is on the other, then by sort of inverse logic, the concentration of water is higher on the opposite side than it is on the side with the more dissolved stuff. And so the water will actually tend to move into the cell. On the other hand, if you take your cell and you drop it into the ocean, where you have a whole lot of dissolved solute outside the cell, then water is going to tend to move the other way through the aquaporin. And this is the concept of osmosis, of osmotic pressure, the net tendency of water to move in a particular direction. And everybody's heard of osmosis, right? Everybody's heard of learning by osmosis, where you put your textbook under your pillow and you go to sleep, and somehow the knowledge is supposed to just morph into your brain. Yeah, you could say it's a rational analogy, going from where there's a high concentration of it in the book to a low concentration in your brain, but it doesn't seem to really work very well. However, with water, it definitely does. And there are three terms that you hear a lot. We're not really going to illustrate them here, but hopefully most of you guys know them. Isotonic, hypertonic, and hypotonic. What does it mean when we say a cell is isotonic with its environment? Yes? Very good. Did everybody hear what she said? There's equal concentrations of solutes in and out of the cell. Another way to think of it is there's equal concentrations of water in and out of the cell. Therefore, there is no net tendency of water to move in either direction. It moves in both directions, but the concentration of water and the concentration of solutes stays the same over time inside and outside the cell. Isotonic environments are very common in multicellular animals. We maintain an extracellular environment in our bloodstream and our tissue fluids that's isotonic to cytosol. And so therefore, there isn't any real tendency of water to move into our tissue cells or out of our tissue cells. If you are a cell living in Lake Michigan, chances are there's more stuff dissolved in your cytosol than there is in the water surrounding you, even though it's Lake Michigan and there's all kinds of crud in there. So that particular cell is now in what kind of an environment? Hypertonic, hypotonic. If there's less dissolved stuff in Lake Michigan water than in the cell, it's hypotonic. Yeah, hypo meaning under. So in that case, water is going to want to tend to move through the aquaporins into the cell because the concentration of water is higher in Lake Michigan than it is in the cytosol. And that's a function of the fact that the concentration of dissolved solutes is higher in the cell than it is in Lake Michigan. So water is going to tend to move into the cell. And then if we took our cell out of Lake Michigan and we put it on, the 9 p.m. flight out to the coast and dropped it in the Atlantic Ocean, it would now be in a hypertonic environment where the concentration of dissolved stuff outside the cell is higher than it is inside. And water, in that case, is going to want to tend to move out of the cell. So this is illustrated in this little YouTube video here. And what they've done is taken some blood cells, red blood cells here, white blood cells here, and they've suspended them in a medium. And the initial medium that they're in, you're going to see is isotonic. Then they're going to change it. Watch what happens. They're in isotonic. Now they have changed it to hypertonic by adding some salt to it. Watch what happens. Look particularly at the edges of these cells. They don't look so spherical, so round anymore. They're kind of crinkling in like peas that are drying up. Now they put it back in isotonic. Everybody's happy again. Now they put it in distilled water, and they misspelled distilled there, so ignore their spelling. Hey, man, our red blood cells disappeared. What happened? Did they go to Narnia? Watch the white blood cells, and you'll understand. Like popcorn kernels. So, dealing with osmotic pressure is actually an issue for many types of cells. And they have to have a way to handle it. Now, if you're not part of a multicellular animal, that is, you're a tissue in the liver or something where you're bathed in an isotonic environment all the time, then you got to have some other kind of strategy. If you're a plant cell, that strategy is to have a mechanical barrier, a cell wall around your membrane. And the plant cell wall is rather thick. It's made of polysaccharides, cellulose and pectin, not pectin, excuse me, pectin and amylopectin. And these polymers essentially act as a buttress against osmotic pressure. The plant is hopefully living in a happy place in some nice soil with lots of organic nutrients, but it needs lots and lots of water. And so the rain comes down, or you, the loving plant owner, remember to actually water the poor little guy, and lots of water gets taken up into the vasculature of the plant, and now the cells are being bathed in a hypotonic solution, one in which the concentration of solutes is much lower than it is here in the cytosol. So what happens? Well, the water flows right through the wall here, goes through the aquaporins into the interior, and the cell begins to swell. But as it swells, it encounters that wall. The wall pushes back. The wall has enough mechanical rigidity to resist that pressure. And so instead of bursting like our white blood cells did there, what happens is the cell becomes turgid. It becomes swollen. And that's what makes your leaves unfurl. That's what makes the stem of your plant stand up. It's that internal osmotic pressure. And so plants have a mechanical solution to this problem. Protozoans, like anybody in here now taking 223, taking the lab? Yeah, so you guys have been working with tetrahymena, right? That's a protozoan. And protozoans have a different kind of a strategy. They have an organelle called a contractile vacuole. And this is a membrane-bound compartment that is associated with cytoskeletal elements that are contractile, like muscle. They actually surround this. And these vacuoles have a connection to the outside world, if you will, through the plasma membrane. And so when osmotic pressure begins to build up inside these cells, the water gets transported into this contractile vacuole. The contractile vacuole begins to swell rather than the whole cell swelling. When the contractile vacuole swells enough, that triggers the contractile apparatus, which squeezes the contractile vacuole and expels all the water through its portal to the outside. So that's how the protozoan is able to go live in Lake Michigan or a puddle of water out there on the street or something like that and not explode because it's in a hypotonic environment. Our cells aren't set up with any special features to deal with this because they never encounter a hypertonic or a hypotonic environment most of the time. The body maintains an isotonic fluid environment for the cytosol of these cells. They don't have this issue. All right, now let's talk about the movement of things bigger and more polar than water. We already reached the conclusion that they're not just going to diffuse across a lipid bilayer, so they're going to need protein help. What kind of help are they going to get? Well, there's two categories of integral membrane proteins that are involved in transporting things, and we call them transporters and channels. And you need to know the distinction between them. A transporter is a protein species that has two forms of gates on it. And a gate is going to be a barrier that keeps something from getting through. And so there's going to be two of these in a transporter. The transporter is going to have highly specific binding sites for some particular solute that are saturable. What's it mean to be saturable? You can be saturated, but what does that mean? Well, concentration, if it gets high enough, will lead to saturation. But what does it mean to be saturated? And we're not talking about the organic chemistry version of saturation here. High quantity, you're getting there. Anybody else? If a binding site is saturated, what does that mean? Okay, just enough for it.
Does that mean like you're gonna have to add more solute to people? Add more solute, it's not gonna dissolve.
Okay, we're not trying to dissolve something here. We're trying to bind something, but he hit on it. If you add more, it's not going to bind. Remember protein-ligand interactions. So here's my ligand, here's my protein. Here's the saturable binding site. Once one molecule of ligand binds to it, there's no room for anybody else. And once all the other copies of this protein have somebody bound to it, there's no room for anybody else. That is, you can raise the concentration of the stuff higher and higher and higher and higher in the medium, and no more is going to bind. Okay, so that's what it means to be saturable. And it has to do, again, with the specificity of protein-ligand interactions. There is a binding site that's got a place that can interact with a particular solute, and once it's done that, until you do something with that solute so that you expose the binding site again, you can't bind any more to it. So that's what it means to be saturable. And it's specific, meaning that just not any old solute will do, only the right kind will do. So that's a feature of a transporter. And because you've got to open and close these two gates, and because you've got to bind something specifically to your protein transporter, and then you got to do something with it, the rate at which a transporter can move something across a membrane is much lower than the rate of simple diffusion. Okay, if you compare that with how fast oxygen could diffuse across the membrane, or how fast the steroid hormone could, much, much faster than a transporter could move something. Now let's contrast that with a channel. A channel is also an integral membrane protein, but it has not two gates, only one gate. And that gate has two positions, open or closed. And so the thing is either accessible or it's not accessible at any time. There is some specificity for ions, and channels you are going to find are going to be transport pathways for ions. There are certain kinds of channels like aquaporins that are not designed for charged species to go through, and by charge I mean full charge, an ion. But most of the channels we're going to encounter are going to be ion channels. And there's going to be some specificity for particular ones. You're a chloride channel, you're a sodium channel, you're a potassium channel. And they won't tend to let other types of ions get through, although there's some slop to that. If you're in the same period of the periodic table, you might be able to sneak through a channel that was really meant for somebody else. And we actually exploit that in the lab sometimes. But it's not the same kind of specificity we encountered over here with the transporter. Moreover, these things are not saturable. So rather than thinking about a ligand-solute interaction like we did with the transporter, think instead like a doorway. As long as the doorway is open, anybody who's the right size, and in the case of ions, the right charge, can get through. And can get through in either direction. So one of the things that implies about channels is that they're not unidirectional. Which way the particular ion species goes is going to be a function of what the concentrations are on either side of the membrane. Whereas with a transporter, transport may be directional, but it's going to tend to only work in one direction. That is, if you've got concentration high on this side, low on this side, and the transporter's in the right orientation, stuff will go in. If you somehow reverse the concentrations, nothing happens. Doesn't go back out again the other way. Although in some cases they do. Depends on the transporter. Last thing about channels that differentiates them from transporters is that because of the way they work, the rate of things moving through them approaches the limit of diffusion. And again, think of an open doorway. It's kind of like, imagine you're standing on the L platform, right? Door opens up, people start rushing in. Basically, as long as you don't get so many people trying to squeeze through that doorway at once that they can't all fit, everybody's moving in and off pretty fast. So it's not quite as high as it would be if there were no door there at all, if there were just open space. But it's still pretty fast. So the rate of movement through a channel is going to approach the limit of diffusion. That is, how fast could things move at a given temperature if there were no barriers in the way at all. There are distinct sets of transporters in different membranes, and this schematic is not intended to illustrate particular transporters, but just rather where you find transporters in cells. And we're going to focus on eukaryotic cells here because they have organelles, which makes them inherently more interesting. But same rules apply to prokaryotic cells. It's just that their transporters are all going to be found in their plasma membrane. And you find all kinds of transporters in the plasma membranes of cells. You find transporters for nucleotides and sugars and amino acids. You find channels for ions there. But you also find some of these things in the internal membranes of the cell. So, for example, mitochondria have transporters in their inner membrane here, for example, for the alpha keto acid pyruvate. There are transporters for ADP and ATP. There are transporters in the outer membrane of the mitochondria, too. There just aren't any shown here. There are transporters in lysosomes, these degradative organelles. And some of them move ions, some of them move solutes of other types, polar molecules, nonpolar molecules, depends on what kind of transporter it is. But every membrane has its own characteristic set of transporters. And sometimes we actually take advantage of that in the lab. If we want to try and purify a particular type of membrane, we say, well, we know this is very highly enriched in this transporter. We can just go looking for that transporter. And the fraction of our membrane that has the most of that transporter in it must be the one that has the membrane that we're interested in. We can classify transport in different ways. One way is according to the direction in which solutes move. Simplest type of a transporter is a uniporter. In a uniporter, one type of solute can move through the transporter in one direction, and there isn't any other type of solute involved. And so uniporters are transporters that carry out uniport. They move a single type of solute in a given direction. This says nothing, by the way, about whether it's active or passive. This is just about the direction. Another type of transporter is called a symporter. And in a symporter, you move two things at once in the same direction. Sym means together. And so you would have a solute and some other solute. Maybe one of them's a small organic molecule and one of them's an ion. Could be. There are all kinds of examples of these. But they would both have to bind to the transporter at the same time, and they would both be moved in the same direction. So that's symport. The last one is just the inverse of this, the antiporter. Two or more solutes moving through the same transporter in opposite directions. So anti meaning against. So we have one solute coming from this side and going this way. Another co-transported molecule is also passing through the protein, but it's going in the other direction. And you can't move the one without the other. That is, there are saturable binding sites for both of them. And usually these things work in kind of a stepwise manner. That is, the first solute binds to a site. That induces a conformational change in the transporter, which exposes a binding site for the second one, and that allows it to bind on its side of the membrane, and only then can you finish the process and change the conformation so you eject the one on one side and the one on the other. So uniport, symport, antiport. Make sure you know those guys. Now let's talk about active and passive. Active means that the transporter is actually using energy to do work. And the work it's doing is moving a solute against its concentration gradient. Okay, now we're seeing something very different than diffusion. Remember, diffusion was a random process. Things tend to move from high concentration to low. In active transport, you're going to move something from a low concentration area to a high concentration area. Spontaneously, it wants to go back the other way, but it can't get through the membrane. And it can't get through the transporter because there isn't one available for it. But because you're trying to get something to move against its natural tendency to diffuse in one direction, you're going to have to spend some energy. And in active transport, that energy can either be expended by the transporter that's doing the transporting, that's called primary active transport, or it can be expended by a different active transporter that's moving something that's creating a source of potential energy that's going to let another transporter move something against a concentration gradient. You contrast that with passive transport, which is something that can happen either in transporters or in channels, but it is going to be entirely diffusion-based. That is, solutes are going to move in the direction that they would diffuse spontaneously. So they're going to move from the side of the membrane where their concentration is higher to the side where it's lower. And all the transporter or the channel does is act as a conduit. It acts as a pathway for that solute to move through. If it's a channel, the single gate is open, and whoever can fit through can go through in the direction determined by diffusion. If you're a transporter, you're still driven by diffusion, but remember, you're going to have a specific saturable binding site. Therefore, you'll let some particular solute or solutes go through you, but not just anybody who happens to be around. Now, let's look at something that's a little harder to wrap your mind around, and that is membrane potential and electrochemical gradients. This is going to get really important when we talk about the function of nerve cells. Cells are chock full of ions, and they're chock full of polar molecules that have partial charges. The environment outside of a cell is also chock full of ions and chock full of polar molecules. However, at any one time, if you look at a cell, you're going to tend to find that there's a little bit more positive charge on one side of the membrane than on the other. And in animal cells in particular, but in cells in general, what you typically find is there's a little more positive charge outside the cell than there is inside the cell. Now, from the point of view of an electrician, what this means is that there is a potential across a membrane. Does anybody know how you define electrical potential? Somebody that's actually into electronics? It's the direction that a test positive charge would tend to move if it had a pathway to move. And so when we say there's a potential across this membrane and there's an excess of positive charge outside relative to inside, what does that mean? It means if we opened up some sort of a pathway and we dropped in a positive test charge, which way would it tend to go? From outside to in or inside to out? Tend to go from outside to in because there's more positive charge outside the cell than there is inside, And therefore, it's just like any other kind of diffusion. Things are going to tend to move in the direction from which their concentration is higher to the direction where it's lower. Now, we illustrate membrane potential by using positive and negative signs. And this is where people sometimes get messed up, because they see that and they say, oh, the outside of a cell has all positive charges on it, and the inside of a cell has all negative charges on it. And of course, that's not true. There's a mixture of positive and negative charges outside the cell, and there's a mixture of positive and negative charges inside the cell. However, there's an excess of positive outside relative to inside. And so we say that the cell membrane has a negative potential, and that's illustrated here with my little voltmeter here. Typically, in a cell, the potential across the plasma membrane is going to be anywhere from between minus 20 to minus 200 millivolts, or thousandths of a volt. It's a very small potential compared to something like the potential in the electric system of a building, where we're talking about 120 volt potential or 220 volt potential. Pretty small, but it's a potential nevertheless, and it means that things that have charges are going to tend to want to move in a particular direction based on their charge. Things that are negatively charged would have a tendency to want to go out of the cell. Things that are positively charged would have a tendency to want to come into the cell based simply on their charge. But it's not all about the charge. It also is about what kind of chemical species are you and how much of you is there inside versus outside. So if you're a sodium ion, you're positively charged. That means that you would tend to want to move into the cell. However, how much sodium is already in the cell versus outside the cell? Depends. And so electrochemical gradients simply refer to the sum total of the electrical potential across the membrane versus the concentration potential. And you can look at them independently. You can say, well, the concentration of solute A is this out here and this in here, therefore it has this much of a tendency to move in. But then you also have to take into account, well, what's its charge? And you have to add that contribution in. Now, if the electrical potential and the chemical potential are both directed the same way, if we had something positively charged out here and its chemical concentration was much lower in here, it would really want to tend to go this way. If there were a channel or a transporter through which it could move, it would want to do that. However, we could take the same solute in a different cell, different set of circumstances, still got the same kind of electrical potential, excess of positive outside relative to inside, potential of between minus 20 and minus 200 millivolts, right? But now the concentration of our chemical species is much higher here than it is on the outside. So the electrical potential and the chemical potential are directed opposite to one another. You still have to sum the two terms up. And so what this means is that while the electrical potential, positive here, says, I want to stay on this side of the membrane because it's more negative in here. I don't want to go this way. Concentration potential says, no, there's a whole lot more of me in here than there is out here. I want to go that way. You put them together, and you end up with this kind of a vector. So electrochemical potential is going to ultimately determine which direction a particular type of solute is going to tend to move through a channel or through a transporter that has a saturable binding site for it. Now let's talk about passive transporters and how they work. So passive transporters, by definition, don't use energy. Therefore, things are going to move in the direction they would diffuse, from high concentration to low concentration. Whichever side of the membrane has the high concentration on it, that's where the solute is going to come from, and it's going to tend to want to go toward the other side. How does the passive transporter facilitate this? Well, again, it's a transporter, so it's going to have a saturable binding site. So let's imagine glucose. Glucose is a molecule that cells can use as an energy source. In fact, they're very good at using it. They will use it in preference to many other types of fuel molecules because they get more bang for their buck out of it. And so they're biased to use it first. Many cells have passive transporters for glucose in their plasma membrane. And in an environment such as your digestive tract, where the concentration of glucose from your food is higher outside than it is inside, these glucose molecules will bind to saturable sites on passive glucose transporters in the membrane of your intestinal epithelial cells. When this binding event occurs, it's going to trigger a conformational change, a shape change in the three-dimensional structure of that transporter. And that happens as a result of non-covalent interactions forming between the glucose molecule and the protein. That's going to close the first gate in the transporter. Remember, this transporter's got two gates. It's a transporter. It has to have it. So you close the first gate. Now nothing can bind until something's done with this glucose. The closing of the gate triggers another conformational change, which opens the second gate on the other side of the membrane. And the glucose is able to diffuse out into the interior. Now, over time, if you have high glucose out here and you keep taking it into the cell, you could imagine that you'd reach equilibrium. You'd eventually get so much inside the cell there wouldn't be a net tendency of it to move in. But that never happens. Now, if you're eating, you know that you don't eat continually. You're not constantly bringing new glucose in. So the concentration of glucose in your digestive tract must drop. So why doesn't the situation reverse and the glucose start coming back out of the cell into your intestinal lumen? Can anybody think of a reason? Yes? Da-da-da-da. Yeah, the glucose doesn't just sit there once it gets in the cell, it gets used. And in fact, the glucose that's absorbed through your intestinal epithelium doesn't, for the most part, stay in that cell. It gets transported out the other side of the cell into your bloodstream. So the glucose doesn't stay there. Furthermore, it doesn't stay in the form glucose. As soon as it enters the cell, it becomes enzymatically altered in a biochemical reaction. Then it's not glucose anymore. It's not glucose anymore from the point of view of the transporter. The transporter can bind glucose, but it can't bind glucose 6-phosphate, which is what the glucose becomes as soon as it gets into the cytosol. Therefore, the concentration of glucose is always lower inside the cell than it is outside, meaning that transport is always going to happen in this direction in your digestive tract. So the transporter is just using the potential energy of the concentration gradient. It's using the energy, but it's not actually transducing it. The concentration gradient is simply causing the solute to move in one particular direction. And if you made this concentration gradient disappear somehow, if you collapsed it, if you literally made the concentrations of glucose on the two sides of the membrane the same, could this passive transporter continue to move glucose in one direction? No, it could not. No, it could not. Now, membrane active transporters are going to do something completely different. By definition, an active transporter uses energy and it moves things against the concentration gradient. And so if the concentration gets higher or lower outside the cell, doesn't really matter. The active transporter is going to use energy to move things in a particular direction. What this figure is all about is what forms of energy can active transporters use. And there are three major ones. The big one is ATP. Active transporters can often hydrolyze the terminal bond in an ATP molecule, lop off that gamma phosphate, release the energy that was stored in that bond. And a lot of free energy is contained in that bond. If you hydrolyze it, you have a very negative delta G. They can use that energy to actually do work. And the work is going to be picking up solutes on one side of the membrane and kicking them out on the other side against the concentration gradient. Now, the active transporter can be a uniporter, it can be a symporter, it can be an antiporter. It doesn't matter. That has nothing to do with the use of energy. So you can have active symport, active uniport, active antiport. Active antiport is illustrated here. We have a real protein called the sodium-calcium exchanger that will bind to sodium on one side of the membrane and move it to the other side while it moves calcium in the opposite direction. And this is an example of a secondary active transporter. It's not actually hydrolyzing ATP. Rather, it's relying on a gradient of sodium that's created by another active transporter, the sodium pump, which is shown here, that allows it to then move calcium against its concentration gradient. And so the sodium pump is kicking sodium out to here, and the sodium concentration, because of the activity of that pump, is higher on the one side of the membrane than the other, so it wants to diffuse the other way, and the sodium-calcium exchanger gives it a pathway to do so. It uses the potential energy in the sodium gradient that was created by the sodium pump to then move calcium against its concentration gradient. So we have a primary active transporter here, a secondary one here. The other type of energy that can be used in addition to potential energies of gradients and then the high energy bonds of ATP is light energy. And there's not a lot of light-driven pumps in nature, but there are some, and they're very important. And one of the first to ever be studied and understood in any detail was called bacteria rhodopsin. It's a membrane protein that is found in certain types of photosynthetic bacteria, and it uses the energy from light to actually move protons against their concentration gradient. And light, just another form of energy. Okay, you have chemical energy, you can have radiant energy, you can have all kinds of energy. Light is just one form. And bacteria rhodopsin and other light-driven pumps are able to capture this energy, change it to a different form in which work can be done to move a solute, in the case of bacteria rhodopsin as protons, against the concentration gradient. And so the electrochemical gradient says protons go this way, but bacteria rhodopsin says, I'm sending you this way. I'm sending you against your diffusion potential, and I'm spending energy that I got from light to do that. Primary active transport, as I said, means that the transporter doing the moving of the solutes is also the one that's capturing the energy. So bacteria rhodopsin would be an example of a primary active transporter. So is the sodium pump. The sodium pump is probably the most important primary active transporter in your cells. And the way it works is that it picks up on one side of the membrane, specifically the cytosolic side, 3 sodium ions. Then, having bound them, changes its conformation to open its second gate, which allowed 2 potassium ions to bind. And potassium has a higher concentration outside the cell than inside the cell than out. And then hydrolyzing ATP, this is going to move potassium into the cell while it's moving sodium out of the cell. Both of these moving against their concentration gradients. Potassium is kept high inside, sodium is kept high outside. Now, you've got 2 species moving, so this is going to be what kind of directional transport here? 2 species moving opposite directions. Antiport. This is a form of antiport. And again, even though the simplest schematic is a 1-for-1, here we have a 3-for-2. So the number doesn't matter. It's still antiport because things are moving in 2 different directions, 2 different kinds of solutes here. So what the sodium pump does, or by its proper name, the sodium-potassium ATPase is it continually creates a sodium-potassium gradient in cells, where sodium is high just outside the cell membrane, potassium is high just inside the cell membrane. Each of those gradients is a source of potential energy that can be used to do work, and it's captured by those secondary active transporters that are able to bind either potassium ions on the one side of the membrane or sodium on the other, and allow them to move to the other side down their concentration gradient. That energy then can be used to bring something else in the opposite direction against its concentration gradient. So here are some secondary or coupled active transporters. And so we've got our sodium gradient created by the sodium pump, and we now have this hypothetical transporter for glucose. Before, we looked at a glucose uniporter, a glucose transporter that only recognized and bound glucose and let it move down its concentration gradient. Now we're going to be looking at a symporter, something that's going to move two solutes in the same direction. One of the solutes is going to be glucose. The other one is going to be the sodium. And the sodium has a high concentration out here because of the activity of the primary active transporter, the sodium pump. That sodium has a concentration gradient that is going to lead it to want to diffuse in this direction. And this coupled transporter, this secondary active transporter, gives it a pathway. So the one gate opens, that allows a sodium ion to bind. Only when the sodium ion has already bound can a glucose bind. When the glucose binds, that closes the first gate. That conformational change then allows the inner gate to open, and now both the sodium ion and the glucose can diffuse out, and you're like, aha. But you're letting all this sodium back in. You're collapsing your gradient. The whole thing's going to stop after a while, right? It doesn't. Why not? Why wouldn't it stop after a while? Yes? Gets used by who? No? We're honing in like a laser. We're focusing in on our secondary active transporter. Is that the only transporter in the membrane? Nah. The sodium pump is there too. And as soon as the sodium comes back in, the sodium pump says, hey, what the hell are you doing in here? Get out. Hydrolyzes some ATP and kicks 3 more sodiums out. And so the sodium pump continually maintains this gradient. As sodium is used for secondary active transport, the primary active transporter, the sodium pump, kicks it back out again, and it maintains that gradient. There's a toxin in nature. It's called wabane. Wabane is a really wicked poison. It's used by certain indigenous tribes as a hunting poison. They put it on spears and arrows, and when they go out hunting boar or small game, they actually use this to paralyze their prey. Wabane has one specific protein it binds to. It's the sodium pump. And when it binds to it, it blocks its activity. If you block the activity of the sodium pump in a cell, what's going to happen? You will not have enough energy to do what? To bring other things in through secondary active transport. So many secondary active transporters are dependent on the sodium gradient. If you stop the sodium pump from working, all of these other secondary active transporters shut down. Not because there's anything wrong with them, but because there's no sodium gradient anymore. And so they can't move things now against their concentration gradient. Cells lose the ability to bring in all kinds of important solutes, glucose, many other things, and they begin to die. So that is what makes wabane a very potent poison. It also makes it a really good lab tool because we've never found anything else in the world it'll bind to except for the sodium pump. So it's really easy to track the sodium pump by using wabane. There are a lot of important, oh yeah, I'm sorry. Mm-hmm. Yeah, it does. And the two examples that I've shown you also illustrate another point. There's more than one way to fry an egg, if you will. There's more than one kind of glucose transporter in living organisms. There are multiple types. And sometimes you have two different types in the same membrane. Other times it's predominantly one particular version or the other. It's the passive version or it's the active version. When we look at absorption through the intestinal epithelium, what we find is that there is secondary active glucose transport going on the side facing the intestinal lumen, and then there is passive glucose transport going on the other side of the cell membrane. Yeah? What could break it down? Well, there isn't a specific enzyme that will do that. And the other thing is that once Wabane is bound to the sodium pump, it doesn't tend to come off. However, remember that cells have lots of copies of the sodium pump, and it can make more. And so if you got a really small dose of Wabane, for instance, eventually the effects would wear off as long as you didn't add any more Wabane, because the cell would make more sodium pump, and that wouldn't have Wabane bound to it. But if you get a big enough dose, you're not going to make it to that point, is the short answer. And of course, if the hunters catch you, you know, it's like, well, you're going to be bacon for breakfast. You're never going to have a chance to recover. But yeah, you can eventually get by it through your capacity to make more sodium pump. So there is a lot of sodium pump found in the plasma membranes of many types of cells, both animal and plant cells. There are then secondary active transporters that rely on the sodium gradient created by the pump, but there are other pumps too. There are proton pumps. Some of them are found in the plasma membrane of cells. Some of them are found in internal membranes, like the vacuoles of plants and the lysosome membranes in both plant and animal cells. So lots and lots and lots of primary and secondary active transporters. One good place to find multiple active transporters, both primary and secondary, is in your kidney tubule epithelium, where, anybody know what's going on in your kidney? What's your kidney controlling? Yeah. It's controlling absorption of ions, and it is specifically keeping your osmotic balance where it ought to be, and also keeping your blood pH where it ought to be. And so there are all kinds of transporters for moving different ions, and there are transporters for moving protons, a type of ion, which of course is what your blood pH is all about. And so in the side facing the lumen of the kidney tubule, you will find a primary proton ATPase that just kicks protons against their concentration gradient out of the cell. There is a proton potassium ATPase that's an antiporter that moves protons this way, potassium ions this way. There is a chloride bicarbonate exchanger, which is a passive transporter on the opposite cell membrane that is moving the counterion of the bicarbonate buffer system out of the cell in exchange for chloride. This is maintaining the pH in the cell environment where it ought to be. So all kinds of different transporters found in the membranes of cells, and they are working together to help maintain a particular set of conditions, a sodium gradient, a potassium gradient, a pH balance, whatever it might be. And that brings up the idea of transcellular transport. Cells that are linked together to form an epithelium. Anybody know a good definition for an epithelium in a multicellular organism? What's an epithelium? Okay, it's a covering, right? And physically, what does it look like? Is it a big ball of cells? What is it? It's a long sheet, very good, sheet of cells. And those cells are linked tightly together. And they form collectively a barrier and it's a barrier that keeps things from moving in one direction or the other unless they've got a pathway not around the cell, but through the cell. And the pathways through the cell are provided by different transporters and channels. And you will find different sets of transporters on one surface of the cell than you will on the other side. And these work in a complementary fashion to allow certain solutes to move across the epithelial layer in a given direction. So we talked about glucose-sodium symport before. That's a form of secondary active transport driven by the sodium gradient. That will bring glucose into intestinal epithelial cells. But then there is a passive glucose transporter on the other surface of the cell called the basolateral side. So you're bringing glucose into this cell faster than the cell can actually use it up, convert it to something else. And that excess glucose can bind to this passive transporter and be moved out into the extracellular fluid where the concentration of glucose is much lower than it is here in the cell. And so you set up this vectorial transport where you go from the lumen of the gut through the cell, out the other side, and then you've got blood capillaries down here bathed in the extracellular fluid, and they've got their own glucose transporters. They pick it up from the extracellular fluid, they move it into the bloodstream, and then it gets dispersed to be used by all the different tissues in the body. And so this kind of polarization of epithelial cells is the norm in multicellular organisms, and not just in animals, in plants too. And it allows specific solutes to be moved from an environment where they are available into a portion of the organism where they are not normally available without the function of these transporters. So here's what you should know about transport and transporters, what diffusion means, what can and can't get across a lipid bilayer. Understand that transport proteins can be subdivided into the transporters and channels. You need to know what makes them different from each other. The three forms of transport based on the number of solutes and the direction they're moving. What distinguishes active and passive transport? What's an electrochemical gradient and how does it influence passive transport? How do the gates of passive transporters work? Remember, conformational change. You bind the solute, that causes one change. The first change causes the second. You open the second gate. What forms of energy can drive an active transporter? We talked about ATP, light, and then the potential energy of solute gradients. And then finally, how do you distinguish primary and secondary active transport? How does the sodium pump work? Why is the sodium pump so important? Because it creates that sodium gradient that all of the passive transporters need. Proton pumps can also drive secondary active transport and regulate cellular pH, and then you can move solutes across cell layers by having different sets of transporters on the different sides of the cell. And so we'll stop there and finish up talking about transport next week.
Of the exam will be posted?
I can't tell you for sure. I mean, the Santrons were just run yesterday because they just reopened the testing center. Okay. We